Thank you, everyone, for joining us for this wonderful conference. This is the first conference for the year for VJON. We've done multiple conferences over the years, and today we are sponsored and supported by Molecusan. So we really appreciate their support, and I'll be sharing a few words or ideas from them as we go through the conference. But what we've got at the moment is a very serious situation from a scientific point of view, where the virus is still circulating, it is still doing damage, and there are not enough people trying to find answers. What we've got here is a group of global experts sharing ideas, sharing their research, and critically, towards the end, after each speaker has their presentation, they will be discussing it with the panelists. After that, each speaker in turn, and then at the end, we'll have a roundtable discussion, which can sometimes be the most valuable thing. And at that point, we can also answer some of the questions that you have. So I appreciate all the people who are already here with us, and we look forward to sharing with you wonderful ideas about what is going on and what can be done. So without further ado, I'll get all the speakers to just say a quick introduction for themselves so that we get to know who they are. And so we'll probably start with Bridget since she's going first, and then Rezia, then Sabine, and then we'll get our two panelists, Shankara and Marcus. So Bridget, just go ahead and say hello to our audience.
Yeah, hello all together. I'm happy to be here. My name is Brigitte König, and I studied biology, chemistry, and medicine. And I'm a professor of medical microbiology and infectious diseases. And in addition, I have a small company which does specific analysis in direction of mitochondria and infectious diseases.
Wonderful, thank you. And Rezia?
Hello, everyone. I'm Risha Pretorius. I'm from Stellenbosch University in South Africa. I'm also affiliated to the University of Liverpool, and I am a physiologist and work in the field of coagulation and microclotting. And of late, long COVID is one of my main focus areas.
Wonderful. And Sabine?
I'm Sabine Hazen. I'm a gastroenterologist by trade. I started my practice as a GI doctor and kind of went into the clinical trial route of doing clinical trials for pharmaceutical companies. My forte in GI and my expertise was in fecal transplant, intestinal microbiota transplant. When that showed a lot more than improving C. diff, I got curious and opened up Progenobiome to understand the microbiome and disease. And I'm spearheading 61 clinical trials on the microbiome and disease. When COVID hit, we were in the right position as a CRO and as a gastroenterologist and as a genetic sequencing lab to kind of understand COVID. We found COVID in the stools. And then from there, the world of the microbiome kind of opened up to us. And this is what I want to share with the world, is really the discoveries that I made on the front line of being a physician with COVID, but also transforming myself to a physician looking at diseases like Parkinson's, Alzheimer's, autism, etc.
Excellent. Thank you very much. And Shankara?
Hi, everyone. I'm Dr. Shankara Chetty. I'm a general practitioner from Port Edward, South Africa. I'm a natural life science biologist and have been a frontline COVID doctor. My, I think, sentinel finding was that we're having an immune response with COVID rather than a virus causing all the pathology we're seeing. So, yeah, very interested in what's going to be discussed today. The sentinel things centering around the pathophysiology of what's causing all the long COVID and long vax injuries.
Excellent. And Marcus?
Hello, my name is Marcus Klotz. I am the founder and director of the world's first purpose-built long COVID post-vac and chronic illness clinic. And here in Cyprus, in Larnaca, we're focusing on offering the largest possible German high-tech portfolio of treatments at accessible prices. And what we do is we're working a lot with scientists around the world to make the latest treatments early accessible.
Excellent. Thank you very much, everybody. And as you can see, we've got quite an audience all over the world who are already with us. And I'm sure as we go along, the numbers will rise. So thank you, all of you who are here. I'm seeing all the comments, and as best as possible, I will share them in the discussion if we have time, but mainly probably towards the end. Before we go any further, what we'll be doing next is we'll be having our first speaker, which is Professor Bridget Koenig. And before we start that, we will just hear a quick word from our sponsor. Thank you very much. And we'll get straight into our first presentation. And I will share the screen for you, Bridget. You just have to tell me to move along each slide. I'll ask you again, just in case people were late, just to do again a very quick introduction of yourself. And we will then bring up your slides.
Okay. I'm happy to be able to study biology, chemistry, and medicine, to work interdisciplinary. And I did my professor in medical microbiology and infectious diseases. And in addition, I have a small company, which is called MMD.
Excellent.
Yes, my topic is spike persistence and mitochondrial damage. We focus on long COVID research this evening. The next slide, please. And perhaps we should just go into a little bit terminology, only short. If we have an infection with SARS-CoV-2, we have sure an acute infection, but also we have a post-acute COVID-19 syndrome. It's called long COVID, and it can last for months and years. And on the other side, we have also vaccination-induced problems, acute vaccination syndrome, we know, and also post-acute vaccination syndrome. But next slide, please. It is more complicated because we have combinations. We have combinations that you first have acute COVID-19, and then you have vaccination, or you have also post-acute vaccination problems, and then you have a COVID infection. So we have different combinations, also with different timely combinations. The next slide, please. So there's a lot of witch theories of the pathogenesis of PAX and PAX, also post-acute corona syndrome and post-acute corona vaccination syndrome. And we know it seems that the persistence of spike proteins in tissues or immune cells is the main problem. In addition, or dependent on that, or is disorders of vascular and metabolic control circuits, inflammation of vascular endothelium, disruption of mitochondrial metabolism, coagulation disorders, and so on. And I stay at the moment on the persistence of spike protein in tissues and immune cells. Next slide, please. Next slide. And just a minute or a second to SARS-CoV-2. SARS-CoV-2 has different proteins, and sure, different proteins can also harm our system. But the spike protein is the most important in this moment because it enters into the cells. And the spike protein, in addition, has a lot of receptors all in our organs. Next slide. And here's the ACE2, also angiotensin converting enzyme 2, is the receptor for the spike protein. And you see the quantitative expression is everywhere in the tissues and mainly in the brain, in the heart, also nearly everywhere. You have this expression, and therefore the SARS-CoV-2 spike protein or the virus, the spike protein, can activate or can compromise a lot of cells and tissues, not only one, but a lot of the tissues. Next slide, please. And this is also one of different publications which shows that the spike protein is really distributed all over in the body. It's, a mice problem on the left side, and you see that it also goes into the brain, into the liver, in the heart, also everywhere, like the expression of the angiotensin converting enzyme 2. And on the right side is the publication. It is in humans that really the spike protein accumulates really everywhere, also in the brain. Next slide, please. And this is a publication. It shows that the SARS-CoV-2 proteins, this time it's an infection, also SARS-CoV-2 infection, that the SARS-CoV-2 spike protein can circulate in the plasma, also in our blood, in the plasma, but also in our immune cells, especially in the monocytes. And it can also express in specific exosomes that are molecules or vesicles which can go everywhere through our body. And we see also the messenger RNA expression everywhere in the tissues, but especially the spike protein is able to go through our body in the plasma, in the immune cells, also macrophages, monocytes, and in the exosomes. The next slide, please. And therefore, we focused on detecting the SARS-CoV-2 proteins in the 3 compartments. In the plasma serum, yes, but I think if we have a lot of antibodies, then it is neutralized. We have doing it in the exosomes and the immune cells. In the immune cells, it's monocytes. We know it from tuberculosis or from HIV, that the macrophages try to catch proteins and to remove them. And if you look on this page, on this slide, you see that we have different combination. We have sometimes only the spike protein in the immune cells, and or on the other side, we also have it in the exosomes and in the immune cells. And if we look at the next slide, please, then we see again different combinations. And here we added the nucleocapsid expression. That means if you have a viral infection, normally the virus has different proteins, like you have seen some slides before. It is a spike protein, it's a nucleocapsid protein, it's a membrane protein, and so on, and others. And the nucleocapsid protein is also a very important protein, which is produced in high amount from the virus. And here we looked whether the nucleocapsid protein is also present in the different departments, also plasma, exosomes, and immune cells. And here you see on the left side, for example, that you have the spike protein in plasma and in immune cells, but no nucleocapsid protein. And on the right side, you also see very high concentration also in exosomes, but nothing of nucleocapsid on the bottom of the slide. It's again a very high concentration of spike in exosomes, but nothing of nucleocapsid. The next slide, please. Yes, and now we look what can the spike protein do. We already have seen that the theory is a disruption of mitochondrial metabolism, and we look on the next slide. We just remember what are mitochondria. Mitochondria, I say that are they are organelles. They have a little bit own DNA, which is about 16,000 base pair. And they have an outer membrane and an inner membrane. The inner membrane is like cristae, where we have the production of a lot of energy. But the mitochondria are not using only, are producing only energy in form from adenosine triphosphate or the ATP, and for that, but it's also producing antioxidants. It is anti-inflammatory. It's anti-apoptotic. That means that that the cells are not dying. It is producing heat, yes, and it is using substrates to produce new cells, for example, in defense, new T cells, to produce all the nucleotides, the fatty acids, and the proteins which are necessary. So the mitochondria have a very important function in health and disease. The next slide, please. And here now I show you some publications, a summary of some publications, but there are a lot, which shows that the mitochondrial dysfunction is a prelude to neuropathogenesis of SARS-CoV-2. It's one of a lot of papers. On the next slide, we see the next example. It was a question, but now it's sure. Do compromised mitochondria aggravate severity and fatality by SARS-CoV-2? At the moment, we analyze a lot of mitochondria, and they are very bad condition. The next slide, please. Also, yes, and here it's a summary of the manipulation of mitochondria by SARS-CoV-2. And we don't want to go now in detail, but we see that it is compromised a lot and different. You have a downregulation of interferon gamma response. You have a downregulation of energy production. You have an increase of inflammation. Also in different parts of the mitochondria, the different proteins of the SARS-CoV-2, especially the spike protein, is manipulating the mitochondrial functions. The next slide. And this is also one of a lot of papers which really shows very good that the SARS-CoV-2 spike glycoprotein interacts with monoamino oxidase and impairs the mitochondrial energetics. And on the next slide, we see also a very important paper or publication. The spike protein impairs mitochondrial function in human cardiomyocytes, also mechanism underlying cardiac injury in COVID-19. We know that the cardiomyocytes, also the destruction of these heart cells, is very one leading parameter of long COVID. The next slides. Yes, and that is that also the SARS-CoV-2 spike protein induces long-term transcriptional perturbations of mitochondrial metabolic genes and so on. And that is a really problem because the mitochondria are necessary for us. Without mitochondria, we are dead, we are dying. The next slide. And one point is when our best friend becomes our worst enemy. I just told you that it is very, very important that the mitochondria must function. But what happens when they are not functioning? Okay, they are not producing energy, they are not producing components, but they are becoming our worst enemy. Why? The next slides, please. And the mitochondria, you remember, are from microorganisms. Yeah? And so they have different parameters or structures which are similar or identical to microorganisms. And if these particles or these substrates like succinate, cardiolipin, N-formyl peptide, mitochondrial DNA, and so is coming out of our cells, then our body says there is danger, yeah, and combats against the mitochondria. So, also dying mitochondria are a danger because our body says that is an enemy, that are microorganisms. So therefore I told you that the best friend are becoming our worst enemy. The next slide, please. Okay, you see it is not very easy, and therefore we must measure different parameters for long COVID post-vac from my view. from my view of infectious diseases and from the mitochondria. The next slide, please. Yes, one point is, okay, this is now in German, but I think you will see it. It's first, we must know, is there a persistence of spike proteins? And the most important is really to look at the plasma serum. It's the first, then the exosomes, and then the immune cells. Yeah, that is the first and most important. But we must know more. We go in the next slide. The mitochondrial metabolism. There we have different parameters which we can look, and that is very important to look how is the function of the mitochondria, or what have we to do? Have we to give some minerals, or do we have to remove the dysfunctional mitochondria, or do we have to go into mitochondrial biogenesis? And this test gives us an direction. The next slide. That is also again mitochondrial metabolism, but especially in immune cells, because we must be aware they must be functional to combat also other infections, but also to combat against cancer and so on. The next slide. Yes, then we must look at our immune system, especially at our cytokines. What is the profile? Do we have more pro-inflammatory cytokines? Do we have more Th1, Th2, or Th17 profile? Or do we have a release of cytokines which are coming more from neutrophils and from macrophages? Or are the cytokines also like vascular endothelial growth factor, which might compromise also the endothelium? So we can see from the profile of different cytokines where might be the problems. It's more the neuroinflammation, or it's more from the neutrophils, or do we have in general a problem with the activity of our immune system? The next slide. Yes, and we should not lose the view on the viral activity. Do we have T cells which can combat our against our SARS-CoV-2, against which variant, also which type of SARS-CoV-2, and do we have a lot of neutralizing antibodies, and are they against the old type, also Wuhan variant, or also against the new variants? What is the combination of the different types also of the SARS-CoV-2? The next slide, please. Yes, now I come to the very summary. The next slide. Again, okay, we know SARS-CoV-2 infection, especially the spike proteins, is combating mitochondria, and leads also to aging of the mitochondria. And aging mitochondria we don't like. The next slide, because aging mitochondria are against our immune system, because mitochondria are the powerhouse of immunity. I have now not the time to explain in detail, but without functional mitochondria, we have no immune system, not combating viral infections, not combating bacterial infections, not combating parasites, and not combating cancer. The next slide, please. And we should forget, the failing immune system is the cause of aging. And aging, again, is the cause of viral infections, of problems of long COVID. Thank you very much for your attention.
Thank you very much, Brigitte. What I'll do is I'll add in Marcus and Shankara, and they will then have a few questions for you. And I'll be looking out for the questions from the audience as well to see if we can get some clarity.
Perfect. If you don't mind, Brigitte, I'll start with the first question. Given the extensive damage caused by the spike protein persistence in mitochondrial cells, what would be the best course of action to reverse or mitigate this damage?
Yes, the first thing I think it's good to remove the spike proteins. because without removing, we have always a problem. But if we remove, the cells must also recover, or the organs must recover. Therefore, we should combine a removal of the spike proteins or destroying of them, but additionally supporting the mitochondrial functions, in a broad manner.
Perfect.
Okay. Bridget, we've seen that with the viral infection, you've got intracellular proteins that are made from the ORF sequences that stop apoptosis, stop interferon responses, and that comes from viral infection. And so you get the intracellular viral proteins. But that infected cell triggers the release of inflammasomes and cytokines, and those tend to spread more rapidly. So what I'm looking at is, with viral infection, you've got a limited number of cells that are actually infected by the virus. Is it the cytokine mechanism that's triggered that causes this to rapidly decompensate, rather than the infection from the virus itself which will inhibit certain cell mechanisms that will cause viral persistence? So is it the cytokine release and inflammasomes that trigger the cascade of injury that we see? Yes, spike protein causes mitochondrial damage, and we'll see that with the vaccine, because it perfuses tissue a lot better than a viral infection would, and we'd see a broad diversity of issues with spike protein. But the virus itself, in its own mechanisms, creates these intracellular proteins that block mechanisms to remove it and creates viral persistence. But viral persistence is a slow, long-term process. Is the quick deterioration in clinical presentation the result of the injury to the cell creating inflammasomes and cytokines, which causes it to rapidly spread, as far as the clinical picture shows?
Yes, your question is very, very important. And I'm sitting and I'm also thinking about your question. Also, it's very important. We don't know exactly because we have also often combinations. Sure, if some people are vaccinated and then they have problems. But sometimes the problems start if they are getting infected, after vaccination. And that we see in a lot of people. Sure, some are also ill after viral infection and some are not. So we have different combinations of possibilities. And I think it's because we are very different. Some have chronic diseases, the other have other problems, also with the mitochondria. Therefore, we see a lot of different clinical pictures. And also I don't know exactly really where we start. It is sure the infection or the cytokines can again induce a cascade. But if you don't have functional mitochondria, the immune system is not able to do anything. Or then you have also additionally neutrophils, which release neutrophil extracellular traps or a lot of RANTES and IL-8. I think it's a combination. It's really difficult from my mind.
All right, Bridget. So I have another question. Many functional doctors have protocols for improving mitochondrial function and switching the energy production from anaerobic glycolysis back to aerobic glycolysis with high-dose thiamine, using fat-soluble variants, TTFD, benfotiamine, activated B complex, magnesium and potassium, selenium and molybdenum. Given the extent of the damage to the mitochondria, is this a viable solution, or do we need to remove the spike protein from these cells first?
Like your first question, I think it's both which we should do. Sure, we can start with destroying the spike or removing the spike protein, but if the tissue is need also all minerals to recover. This must go a little bit in parallel. Sure, you can start first with the spikes, but then you have to look at the recovery of the cells. But I think a lot of people say coenzyme Q10, NAD, Nicotinamid dihydrogen, whatever. But we have to do a lot, not only one substance. Yeah, because if you really look at the biochemistry, they all interact each other. Yeah, and if you only look at one, you have problems at another position. Yeah, and then also you must look whether you have perhaps to remove the damaged mitochondria and to go into the mitogenesis, also biogenesis, to amplify or to regenerate the good mitochondria. Also it's not just one substance which you should give. Yeah, it's and then it's also one important is to look which biomarker or which biomarkers show me I'm on the right way. Yeah. Sure, one simple is to look that the spike protein is disappearing or destroying. Sure, that must be, yeah.
Yeah, that's why we keep DHL so busy with sending samples to you every day.
Yeah.
Can I ask an audience question, Bridget? This is somebody's question here. In your testing, do you distinguish between the spike protein from infection versus spike protein from the vaccine?
At present, not.
Okay,
Also wir at MMD not, no.
Just checking if you have any other questions before I look into the audience to see if they have any questions that may be relevant here to Professor Koenig. Here is somebody was saying here, she thinks she is vaccine damaged, but did not have a COVID infection. And I thought persisting spike might be the problem, yet have negative spike protein test results. Any thoughts on that when you have people with symptoms, but no presence or no evidence of spike protein in exosomes or immune cells?
Yes, also first we have to look where did they look. Did they look really in exosomes and in immune cells or only in plasma? If it's really negative in exosomes and immune cells, then there are two possibilities. Either there are also other mechanisms, which could be a reactivation of Epstein-Barr virus, reactivation of herpes simplex virus, or other problems. But we also know that in a very low number, it's not the regular case, that the spike protein or the are not released from the body. So if there's done doing like hyperthermia or nicotine therapy or something else, then suddenly it is appearing in the exosomes, the spike. Yeah. So it could be that the cells are really damaged and they can't remove the spike protein from the tissues, and it can't be in the exosomes and the immune cells. It could be one possibility.
Yes. I've got another question. This is a more direct question from someone who has had long COVID for about 4.5 years. Very high spike on antibodies to the alpha variant, which made them ill. They have had a spike protein test with exosomes. And what should they be considering as a treatment, just generally rather than specifically?
Yes, it's very that you have spike protein also in blood, I think in plasmas. I think then it's a difference, not only one therapy, not only one treatment. I think there's several treatments. Also supporting the mitochondria, looking at some therapies like at the apheresis center in Cyprus. Also different treatments. Not to go in detail now, but I think he or she can contact the other in this group to look at the treatments. Also it's not only one treatment.
Yeah. Another question that was thrown in, I'll let you guys come back in a minute. I thought this was an interesting question. Someone says they've found that hyperbaric oxygen therapy was helpful in treating spike damage and mitochondrial injury. Any opinions with regards to that?
Yes. And there's several studies also from Israel. They show that the hyperbaric oxygen therapy is helping restoring also the telomeres, and which are also in combination with mitochondria. So this might be okay, and I think also in Rostock here in Germany, they are also using the hyperbaric oxygen therapy for treatment. But I never would say it's in general. You have also look really which patient is for which therapy. Yeah, as it's not you have intermittent hypoxia. You have ozone therapy. You have hyperbaric oxygen therapy. You have other types oxygen therapies. You have magnesium therapy. I think every therapy is really good, but we must know, or we have to know, which therapy is the best for which patient. Yeah.
Excellent. Any other questions, Shankar or Marcus?
Yes, if you don't mind. So,
Yeah.
Brigitte, given that the mitochondria are functioning in an anaerobic state and the byproducts of this are lactate dehydrogenase, could this explain the large number of liver failure and renal issue patients that are now experiencing post-COVID or post-vac, given that the LDH is returned to the liver for removal?
We have a little bit to distinguish, or I misunderstood it. If it is produced a lot of lactate. Lactate dehydrogenase is an important enzyme, but it is released if the cells are dying. Yeah. Cells are dying also when the mitochondria are not functioning. Therefore, lactate dehydrogenase is a parameter for dying cells, tissues, for heart attack or whatever. And sure, if you have a high number of lactate dehydrogenase, you have also might be that you have problem also with your liver and with other tissues. Yeah.
Excellent. Any last question, Shankara?
Philip, I just wanted to add some context. We've got two different scenarios at play. When we talk about spike protein, we've seen the biodistribution of spike protein into many tissues. So even if it's not found in the blood, in the plasma, that doesn't mean it's not there. It's in certain tissues in the body. So you still have persistence of spike. The spike protein on its own will damage mitochondria and damage certain cells, which is damage to the cell itself. But persistence in any area of the body can still trigger continuous immune dysregulation. Like the patient said, for 4.5 years with the hyperimmune syndrome. That is persistence of spike, and you might not see it in the blood, you might not see it in the immune cells, but as long as it's persistent anyway in the body, it'll trigger immune dysregulation. It's the peanut in the mouth with the guy that's allergic to peanuts.
Wonderful. Thank you all very much. And we're going to bring in our next speaker now, and we'll be able to discuss some of these points in our roundtable towards the end. Thank you very much, Brigitte, and we'll talk to you a little bit later. Thanks, Shankara and Marcus. Hi, Razia. Thank you very much. And your presentation, I'll just ask you, as usual, to do a very quick introduction, and then we'll go straight into your presentation.
Thank you very much. Just to check, you can see my presentation full screen?
I'll just add it in here now. Yes.
Thank you. Great. Hello, everyone. I'm Risha Pretorius. I'm from Stellenbosch University in South Africa. I am at the Physiological Sciences Department of the Faculty of Science, and I study coagulation and how it changes when you have inflammation. And today I will be sharing with you updated findings on microclotting and inflammation in post-COVID disease. I just wish to acknowledge a whole team of clinicians, research students, and collaborators. There are many more, and that I have not put in this slide, but I would like to acknowledge them individually in the presentation as well, where applicable. However, I need to acknowledge Professor Douglas Kell, my long-standing collaborator. We have been collaborating since 2011. Our lab research will not have been possible if not for amazing funders. I would like to say something about them in this slide: Polybio Research Foundation, and particularly Amy Prowell, Balvi and Kandu Research Foundation, Colonel's Crowdfunding Initiative, and thousands of clinicians, researchers, as well as patients who made it possible for us to obtain our imaging flow cytometer that's in our lab, and then standing up to POTS Foundation and also the South African National Research Foundation and the MRC of South Africa. So over the years, we have published numerous papers that show the importance of inflammatory molecules in circulation and their role in abnormal clotting. We are particularly interested in platelets and platelet signaling and the role in abnormal clotting. Platelets in circulation play a critical role in healthy blood clotting. However, they can become overstimulated and can drive pathological blood clotting if there are inflammatory molecules in circulation. This can also happen in the presence of viral and bacterial infections, where platelets can act as important signaling entities. There is also a complex relationship between the receptors on platelets and endothelial cells, where circulating biomarkers may bind to. In the context of COVID-19, platelets are therefore central in immune activation and general coagulation pathology, and they can form various complexes, and obviously also within cells. This is known as platelet clumping. So over the years, we've been studying clotting, and we are interested in pathological clotting involving the main clotting protein fibrinogen, which is a soluble protein in circulation. Together with Doug Kell, we discovered that these soluble fibrinogen molecules may become insoluble in the presence of circulating inflammatory molecules, and then they take on an amyloid structure. When they interact with the inflammatory molecules or inflammagens from the various viruses and bacteria, this happens. And also when they interact with spike protein when it's in circulation. We have shown this amyloid presence in blood plasma using various amyloid protein markers like theoflavin T and amitrackers. Theoflavin T is a well-known marker of amyloid protein and has been used for many years to study amyloid protein and in particular amyloid beta in the brains of Alzheimer's patients. But we need to note that in blood, this is not amyloid beta. This is a protein changes in fibrinogen, which takes on an amyloid structure. We call these structures fibrinogen amyloid deposits. As I will discuss a little bit later, only in 2021, we started using the term microclots. Here is an example of our earlier work on ischemic stroke. The green signal is that of our marker of misfolded protein, theoflavin T, and it demarcates the amyloid areas in fibrinogen. In the micrographs on the left in A is an example of blood plasma from a healthy individual with no visible amyloid protein misfolding. And then the micrograph on the right shows plasma from a patient who had a thrombotic ischemic stroke. Significant amyloid plasma protein misfolding is visible. Between 2014 and 2016, we discovered that the potent inflammagens that comes from the walls of bacteria called LPS can directly bind to plasma proteins and cause them to become amyloid. Here are micrographs of our earlier work. On the left, you can see plasma from healthy participants with minimal green theoflavin T signal. And on the right in B, where LPS was added in low concentrations, significant amyloid formation was noted. This laid the foundation for our later work in acute COVID and also long COVID. We also studied clotting pathologies in various inflammatory diseases by adding thrombin to the blood or blood plasma. Thrombin is the last step in the clotting cascade that transforms the soluble clotting protein fibrinogen to an insoluble gel of fibrin clot. It is well known, and it's a well-known lab method to study how fibrinogen clots into this fibrin gel. In the micrograph in A, you can see a healthy fibrin net created from plasma of a healthy individual. In the healthy individuals, this fibrin clot looks like a little fish net. But in diseases like diabetes, cardiovascular disease, and stroke, the fibrin clot becomes dense and matted, as can be seen in the micrographs B and C. This clogs up the vasculature and can cause significant pathology. We were also interested in the structure of red blood cells and their death mechanism called eruptosis. On the left, you can see a red blood cell from a patient with severe iron overload disease known as hemochromatosis. Too much iron in circulation causes oxidative stress, resulting in significant inflammatory processes. On the right is a damaged red blood cell entrapped in fibrin from a patient with thrombotic stroke. In 2014, we were the first researchers to show that in Parkinson's disease, there are significant red blood cell eruptoses. This we directly attributed to circulating inflammatory molecules, and at that stage, the idea of a microbiome in blood was still a very new concept. We argued that inflammagens like LPS from bacteria in circulation could also directly cause such pathologies. Now it is widely accepted that there is indeed a blood microbiome and that it has significant impact on pathology and disease. Fast forward to the pandemic. Early in 2020, we looked at blood clotting in acute COVID using scanning electron microscopy. We found that platelets were damaged and clumped together and attached to red blood cells. The fibrinogen and clotting proteins that are supposed to be soluble formed spontaneous deposits. In the micrographs on the far left, you could see platelets that are hyperactivated. Note the yellow arrows. In the middle are micrographs from platelets that are attached to red blood cells. Note the white arrow. And on the far left, you can see micrographs of spontaneously formed plasma protein deposits that even glue together red blood cells. You can see that with the blue arrows. During the pandemic, we did not have access to thrombin. Thus, I decided to only add theophlavin T to our platelet-poor plasma. Here you can see the differences in platelet-poor plasma protein structure in acute COVID patients, where we compared the plasma smears of acute COVID to that of healthy and also to diabetic plasma samples. Over the past months, we have spent time to look at how we can make the nomenclature of microclots clearer. And therefore, we focused now on characterizing different microclot phenotypes. It is not only or not simply the size and the numbers of microclots present in healthy versus long COVID, or then any other disease with circulating inflammatory molecules, but rather their content, meaning the inflammatory molecules of interest, their activity, as well as the biochemical characteristics. Such characteristics make the concept of microclots ideal candidates for phenotypic characterization. We've also been looking at the biochemical characterization of the various molecules that us and others have found entrapped inside the fibrinoid microclots. We know that microclots stain with theophlavin T, Congo red, and amitracus. However, inflammatory molecules associated and entrapped may also have their own amyloidogenicity. By using the website Ameliogram, the amelogenicity of any protein can be determined. Here is an example of alpha-2 antiplasmin, a molecule that prevents clot breakdown. We have found this molecule entrapped inside microclots using proteomics. In fact, all of the inflammatory molecules we found associated with microclots have significant amyloidogenicity in themselves, making them sticky and less prone to attach to microclots as they roll over damaged endothelial cells. And here you can see just an example of a ameliogram site in the diagram that it can generate. Here are also the possible different phenotypes that I and others have identified so far. For example, where fibrinogen form, where it's a main component of the microclot, where it's associated with nets or neutrophil extracellular traps forming immune complexes, or fibrinoid microclots seeding onto cellular debris from damaged blood cells, blood vessels, or also the membranes of blood cells like red blood cells. There might also be a phenotype resulting in heterogeneous inflammatory molecule aggregates together with fibrinogen, but which seed onto red blood cells and endothelial cells that are still intact and are circulating through the body. That these ones might then themselves be pro-inflammatory. So we have previously added spike protein and LPS to purified fibrinogen, and we used fibrinogen alpha chain antibody. And here you can see some of our results. The fibrinogen aggregates indeed form fibrinogen microclots, and this can be one of the phenotypes. Bacterial biofilm can also be found in the microclot complexes, and this was done with one of my students in my lab, Anel, albeit it is in very low signal. Another important phenotype might be the immune complexes, and we worked with Professor Allard Thierry from INSERM France. He's interested in net formation, and we looked at neutrophil elastase, melioperoxidase, as well as circulating DNA. And here you can see that the microclots are structurally associated with nets. We have long said that long COVID is a thrombotic endotheliitis. Here we found endothelial damage molecules shed through the damaged vasculature and entrapped in the patients with long COVID. And then here is a close-up of deposits on red blood cells where microclots seed onto intact cells that are in circulation, red blood cells in circulation, and obviously potentially have a significant impact on those red blood cells' function. One of the questions we frequently get is whether microclotting is related to macroclots seen in thrombotic stroke. Circulating back to our research many years ago, we found significant microclot deposits in blood samples from acute thrombotic ischemic stroke. This research was done in collaboration with neurologists from Pretoria University. At that stage, we were more interested in the red blood cell pathologies and also did not yet call the plasma deposits microclots. However, now looking back at our data, the microclots were hiding in plain sight. I'm happy to report that with Doug Hell and a whole team of researchers, a neurologist of the University of Liverpool, and with Melanie Walker from the University of Washington, we have just published a paper in the journal Stroke, showing that retrieved macroclots from patients that had thrombotic stroke indeed contain significant amyloid proteins, as demarcated with theoflavin T. This suggests that microclots are on the biochemical and physiological pathway to the formation of macroclots, as seen in stroke, suggesting that microclots lead to macroclots under specific inflammatory and pathological conditions. Together with many researchers that is from all over the world and part of the World Health Network, we recently published Delphi, where researchers and clinicians of 28 countries participated to write this Delphi paper. And this forms the initial guidance to address the spectrum of long COVID as a disease and reinforces the need of translational research and large-scale treatment trials. Together with a team of clinicians, we also wanted to give some guidance to clinicians on how to determine if their patients indeed have thrombotic endotheliitis. We wrote this paper entitled Vascular Pathogenesis in Acute and Long COVID, Current Insights and Therapeutic Outlook. I'm very happy to report that it was published in Seminars on Thrombosis in Hemostasis, and we won the most popular paper for 2024 in this journal. We have just pre-printed a paper done with a team of SU researchers and my postgrad student, Maxine Waters. We looked at proteomic signatures of post-vaccine, post-infectious syndrome, we call PVPIS, and discussed insights into immune dysregulation and coagulopathy. We compared the protein signatures of previously published data from proteome analysis of long COVID patients and found that individuals with post-infectious syndrome had distinct protein footprints, suggesting that vaccine injury might lead to a different physiological pathway of activation. In this preprint, I'm very happy to share, and that was just preprinted with a team of Derby and under lead of the Fahy group, found that exercise may worsen symptoms in long COVID due to microclot fragmentation. We know that many with long COVID report post-exertional symptoms, and this may worsen after physical and mental effort. In this study, we attempted to explain why, and we show that microclots fragment after exertion, potentially triggering inflammation and reduced oxygen transport. We have in the past shown that microclots entrap inflammatory molecules. When fragmented, they may release these molecules into circulation, amplifying inflammation and impairing oxygen delivery. The Derby team employed a 2-day regime of submaximum cardiopulmonary exercise testing, or CPET, and we assessed 46 patients. The implications are that if exercise fragments microclots but does not clear them, this may worsen microvascular function. Our paper challenges the rationale for graded exercise therapy in long COVID. Furthermore, it underscores the need for exercise to be carefully considered. Most importantly, the effects of exercise should be objectively monitored and patient managed carefully. Now, I'm nearly done with my presentation, just very shortly. What is happening in the next near future? We are working with various people around the world to look at different microclot populations in their samples that are sent to our lab. We are also working with Jeroen den Dullen and his team, and we have optimized the microclot imaging flow cytometry test in his lab in April, and we will be using full body PET scans to detect microclots in patients and organs. One of our clinical lab collaborators is with Innes Laboratories in Switzerland. They are currently doing the microclot test on their imaging flow cytometry for clinical utility. We have also been looking at the possibility to either isolate single microclots or at least manipulate them with a nanotweezer. And here you can see the microclots manipulated with a nanotweezer. And this was done with Peter Neertling and his whole team, Leroy Duplessis from his lab. And this is a video just to show our preliminary results. Lastly, a team of radiologists from the University of the Free State have recently found abnormalities in the carotid arteries that are atypical. And this team is currently investigating the phenomenon. And looking at it more closely. You can see here the fluffy deposit in the carotid artery in the micrographs kindly provided by this team. And with that, this was a very quick trip through our research endeavors the past few months. And I conclude this presentation. I thank you for your attention.
Wonderful. Thank you very, very much. Appreciate that, Rezia. And before I go any further and bring in the our panelists, I just wanted to take a moment to remember our guests who are with us, who are busy commenting. And I'll be trying to share some of the questions after our panelists have been able to share some their own questions, but I'll be looking out for questions from the audience as well. So let's get straight into our panelists then and see what questions they have to ask.
Perfect. Rija, thank you very much for this. Let me also congratulate you, of course, Dr. Koenig as well. Phenomenal work. Just your last paper, absolutely love it. And thanks for bringing the biocode tests to life with Yannick. Our patients and our clinicians here, we love it. Just the detail that we get now is amazing. Now, the biocode test, the flow cytometer, and the PET scans, is that the future of microclot testing in your eyes?
Yes, definitely. Right in the beginning, we looked at the microscopy. So that is our team's main research focus. We soon realized that microscopy is not useful for large-scale clinical utility. It's easy in a research setting. It is very difficult to do it on a large scale. So we crowdfunded via kernels, and many clinicians and patients contributed. We got our own imaging flow cytometer. We patented the method, and Biocode is distributing it to all laboratories that would want it worldwide. And as you rightly mentioned, Innes Laboratories with Yannick Koch and Felix Kalkmann in Switzerland is now doing the imaging flow cytometry method. It is it's totally easy, it's fast, it's reliable. And what makes it so nice, you put in the sample, you don't search under a microscope. It can't be said that you are cherry picking certain areas. Whatever you put in, you get out, and that is the result. And it's visual because it's the imaging flow cytometer. And so it's an easy modality. The PET scan on the other side, as you mentioned, I work with Jeroen den Dullen. It's a very expensive endeavor. So this is a first attempt to search in the body from the head right through all the vasculature into the organ system on where these microclots are situated and the amount of damage they cause and how, if we can detect them. So we haven't done it yet. However, in his lab, in Jeroen's lab, we have set up the imaging flow cytometry method, the exact same method that Innes is using, so that they can screen patients before they take them to the PET scan to determine how the microclots look in the blood. And then hopefully we have an idea, the severity of the microclots in the blood, how it looks with the PET scan in the body.
It's also a very beautiful bridge, the analysis and the apheresis, because Innes has both of those. And it's just amazing how progressive you and the boys in Switzerland are. Thank you again.
Thank you.
Rezia, you... With the microclotting, we've seen different speeds at which people clot depending on the vaccine, and of course with the viral infection. With the viral infection, people developed microclotting very quickly. It seemed over a few days, and we tracked it with D-dimer tests to see how they progressed. I noticed with the vaccination, patients have a cohort of patients have elevated D-dimers, but not that high as we see with the virus itself, and not as suddenly progressive as we see with the virus itself. As well, we also noticed in the third wave, where we saw a lot of clotting issues, the clotting issues were slow to establish themselves. Now, you mentioned a immune complex mediated kind of clot formation. And in the first wave, it seemed to be more a type 1 kind of reaction because it was very quick from the onset of the immune dysregulation. In the third wave, it was a little slower and a little more difficult to treat. Is the clotting closely tied to immune complexes that might be forming the amyloid kind of microclots, like in a condition like erythroblastosis fetalis, where we should be giving anti-spike antibodies in case you have spike antibodies that trigger immune complexes that might lead that particular cohort of patients to develop microclots, because not everyone does it. Could that be related to blood groups like it is in erythroblastosis fetalis, and show us the cohort that is more prone to developing microclots?
We haven't studied that specific phenomenon to see how it's linked to different blood groups. But what we did realize quite early in the pandemic with the more severe alpha and beta and deltas, those individuals developed microclots that were much larger and much more complex in content, I think, than the later Omicron virus phenomenon. So in Omicron, the microclots were smaller if we look at the acute infection. However, we do find that when you develop Omicron and you develop long COVID, then over time, the microclots are basically, then you can't distinguish. Then we can't distinguish just by looking at the microclot numbers and the sizes. However, you say something very important about the immune complexes. This is the next step, as I mentioned in the presentation as well. We need to characterize different microclot phenotypes. So they are, I think, in different individuals, you might be prone to have a different phenotype depending on your specific immune reaction, whether you've got viral persistence, whether you've got microbiome involved in your disease, and your inflammatory status, and the level of endotheliitis in your, inflammation of the vasculature. So it's crucial for us. And perhaps the next steps also, as Mark has mentioned, with different methodologies to remove the microclots or also to characterize them at INES, is to actually get more clarity on which phenotypes are in which individuals. I do believe that many individuals have these immune microclot complexes, which then you will find nets structurally attached to the microclot areas. And that's very important, because I think if we can figure out which specific phenotype, perhaps there's some people with all of the phenotypes, in the same numbers, but some might have more prone to the one or the other. We need to characterize that. And we think in our lab that we've got 5 types of phenotypes now, but there might in fact be many, many more. The dilemma for us is it is such a new field, and people have seen microclots as debris or non-specific or just rubbish. You don't need to look at it, you can just ignore it. But it's now becoming clear that this part of ignored structures in the blood is actually of significant importance, and to remove it, but also to prevent it from happening, and very important to identify in patients which ones are more prone.
Can I ask, Razia, this is a question that came from the audience here, from Tom. Clinical genetics may reveal a predisposition to being vulnerable to microclots, which is fitting in exactly with what you were just saying.
Absolutely. And I just watched the space as well. I mentioned that we had this paper in preprint with Maxine Waters and Professor Marita Kotze. She's from the University of Stellenbosch as well. We had funding from the South African MRC. In this project, Prof. Marita Kotze's team, she's a geneticist and she specializes in finding the different genes that are associated with coagulation dilemmas. And that is spot on. We do believe that there is a population of individuals with genetic predisposition to be more prone to clotting. And perhaps that could be a part of the test. But more importantly, in future, if there are more vaccine rollouts, one could argue that patients could perhaps, before they get the vaccination, to be able to see if they've got a genetic predisposition for clotting. And then perhaps such individuals should be very careful not to take a vaccine that can cause clotting. So we're in the process of teasing this out. I know many other groups all over the world is looking at various genes to identify which ones play roles. But clotting and genetic predisposition are central in what we have been seeing.
Philip, Reza, can I add a little clinical experience there? I've been treating patients with Omicron, and I see the reactions in both immune and coagulation that get triggered as the illness progresses. I've never had anyone really severely ill with it, but we do the D-dimers and things like that, and we suspect if there's microclotting, we look for it and treat it accordingly. But just my case in point, I've never had the three variants of the first three variants of COVID, but when I had Omicron, on both occasions, I started to clot 1 week into the illness, and it occurred within a day, from no symptoms to ICU. And on both occasions, if you look at it, I considered it almost an immune priming. That's why I mentioned immune complex kind of illness. The first time round, I developed it exactly on the 8th day with a loss of smell, and the next day, my D-dimer went up to 800 from should be below 500. I was anticoagulated. The second infection, exactly the same on the 8th day, developed double vision. And on the following day, again, microvascular clots. But this time around, my D-dimer went up to 6,000, the second episode. So I've had to stay on anticoagulants chronically. But then that draws my attention to the fact that all of us don't respond to it the same way. And my family, the boys, my cousins, my father's brothers' sons, all developed microclotting when they had COVID. Yet I've had other family members that are not on the father's side of the family or males that didn't develop the microclotting. So there's definitely some kind of genetic phenotypic preponderance or predisposition that's there. And I think that's where we need to start to try and sift through who's going to react in which particular way, seeing the diversity of reactions we see.
Can I bring in another point that was made here? This is from Pat, who was saying that in her patient population, the numbers on anticoagulation, apixaban rose threefold between 2020 and 2024, and for rivaroxaban, another anticoagulant, fivefold in the same period. Based on the principle of the microclotting, is it now going to be that anticoagulation is an essential part of longer-term management?
That I cannot answer, but what I do know is that we have seen, I think, worldwide a rise in cardiovascular type dilemmas and disease. What I also, if I just can circle back, Shankara, to what you said, one of the important things that we have noted is in some people with long COVID who do have microclots, when we look at the microscope or with the imaging flow cytometer, they do not have a high D-dimer. So D-dimer we found is not a good test for microclot breakdown, because some of the microclots inside them have got molecules that prevent clot breakdown. Interesting, some of our clinical collaborators have seen if they then give anticoagulation like apixaban, then the D-dimers suddenly go up as the microclots break down, or in some cases even nattokinase, which is like a nutraceutical that breaks down clot. In that cases, then microclots do break down. So it's tricky to say that you've got microclots if you don't have the test to look at it, because in some cases obviously D-dimer is a very important marker, but in some people it simply is not shown.
Excellent.
So, actually, remember in the days when I got the first size microscope, the fluorescent one in Germany, we had numbers of 100% of the samples you tested were in long COVID cases were showing microclots, and about 80% in ME/CFS. Obviously, now you have been going through a much more broad patient population. Do you have any new numbers to share? Long COVID, ME/CFS, and other illnesses?
Yeah, so if we test long COVID patients with a diagnosis, so someone must have been new onset symptoms after acute infection, you know, the typical long COVID profile. We have seen microclots in every single patient that we have looked at. It might not be as high as with alpha and beta and delta variants, but we do see microclots if they've got complaints of long COVID symptoms. If they do have, if the clinician feels that they've got vascular complications or endotheliitis, we do see microclots in those patients. Perhaps not as high as previously seen with the earlier variants. If we look at ME/CFS, they slap bang in the middle. So if you've got your control here, your ME/CFS is raised microclots, and long COVID is still more. So it is, it's halfway between the two. But on that note also, we also see significant platelet hyperactivation in ME/CFS and long COVID, to such extent that if you look at the platelets of these two populations, you cannot distinguish between the two. Definitely, they've got less microclotting. It might be due to the fact that ME/CFS, specifically before acute COVID, have got other viruses, so also post-virus syndrome, other viruses that cause or played a part in the disease that perhaps is not so virulent as the spike. But the dilemma is what we also notice: many long COVID patients left untreated goes into typical ME/CFS symptoms, which is significantly cumbersome and worrisome for us, because how do you how can we treat, and we need trials, and that is the most important thing that we need to do.
Excellent, wonderful, and we'll be holding on to you afterwards when we get to our roundtable, Rezia. And what we'll do is move straight on to our third and final speaker. So thank you all very much for that. And so here we go. Thank you, Sabine. How are you?
I'm doing good.
So what I'll ask you to do, Sabine, is to do a quick introduction of yourself and then to go into your presentation. We are trying to hold to 20 minutes
Yes.
and then we'll have our panelists discussion. I'll put up a 5-minute signal for you when we've reached the 15-minute mark. Okay?
Perfect. Just make sure you see my presentation.
Let me just make sure I can see it. If not, I'm not seeing it. Can I use mine and you just tell me to move along the slides? Perfect. Yes. So I've got that ready. Okay. Do a quick introduction and then we go straight into your presentation.
All right. Perfect. I'm Dr. Sabine Hazen. I'm a gastroenterologist that did clinical trials for almost 3 decades. Clinical trials brought me to the world of the microbiome, where I'm trying to understand the microbiome and diseases. Next slide. So next, the topic today is Save the BIF. Disclaimers: I own too many hats. Ventura Clinical Trials, CEO of Progena Biome Microbiome Research Foundation. Next. The island of research. Do not block the path of inquiry. It's all research out there. Next. If we knew what it was we were doing, it would not be called research, would it? Albert Einstein. Very important as we get into the microbiome space. Next. So the microbiome tells the story. Next. What is the microbiome? A collection of all genomes in an ecosystem. Next. The technology we use is next generation sequencing, whole genome sequencing by DNA and RNA. The assay, to the best of our knowledge, is valid, verified, and reproducible and normalized. We are now up to 61 clinical trials, and we believe we know what a normal control is. However, I will demonstrate the challenges of the microbiome. Next. So next. So the challenges of the microbiome, unfortunately, is that we're all different. If you look at the previous slide, you will notice that there's a lot of previous. Thank you. Yes. Each column is an individual. Each color is a group of microbes. As you see from one to the other, they're completely different. So patient 3 has a lot of Bacteroides, patient 2 has a lot of Bifidobacteria, but yet they're deemed healthy. So what is healthy if we're different? Next. So if we're all different, how is there such a thing as a normal? Age, race, gender, BMI, vegan, carnivore, Indian, drugs, past medical history, surgery, all make an impact on the microbiome. Next. So the other challenge is that it's an expensive technology. This is deep sequencing. We do not look at the surface where we look at what you ate last night. We look at what's sitting on the base of your colon. Next. At best, the microbiome testing is a research tool. There's no real clinical data out there. Mostly a lot of mice studies and rat studies, and unfortunately, they do not reproduce on humans. Next. So you have to be very critical of the data out there. The microbiome in specific disease, over $466 million has been spent since 2016. And when you look at the data, 90%, in my opinion, is not validated. If we cannot see it, if we cannot see a microbe that has been deemed a microbe from MS or Parkinson's, and we do not see it in our clinical samples, we have a problem. Next. So the other problem is, what do these microbes mean? When you find Akkermansia in the gut, is that a good bug or is that a bad bug? Is C. diff even a good bug or a bad bug? Is E. coli a good bug or a bad bug? Finding COVID in the stools, is that a good thing or is that a bad thing? Next. So again, we're all different. So how do you make sense of this? Next. You kind of have to look at families. And this is my family portrait. And you'll see the two in the middle are very similar in their microbiome composition. And the two at the ends are very similar in their microbiome composition. I can tell you that the one daughter and me are identical, and my husband and my other daughter are identical in their personalities. Is there a microbiome similarity in families that makes us so similar in our personalities? Next. Now, when you look at families with autism, the first column is a mom. The second column, third and fourth are triplets. You will notice that the first column has a lot of microbes that are apparent in the mom, but the two in the middle did not accumulate so much of that microbe. But the third one, who is autistic, has a lot of that microbe. Is that a signature microbiome that we can say, maybe this is the reason that this kid is autistic? Is autism even a gut dysbiosis? Next. So the microbiome tells the story. I'm going to step into COVID next. So COVID was really the light. The only thing that I could say, the light at the end of the tunnel of COVID is really, it got us to understand the microbiome a little bit better. First, me on the front line, seeing my stool samples on a monthly basis and seeing when I got COVID in my stools, when I didn't, etc. And how did I survive, and how did I not get any antibodies the whole pandemic? That was really kind of, I was the N of one, the guinea pig. But more importantly, we saw a lot of data. Next. We first found COVID in the stools by whole genome sequencing. Next. The first thing that we discovered was that these sequences are all different. This is the whole entire virus, and this is not one or two copies. This is multiple copies. Next. And even at the spike protein, there's a lot of mutations. You'll see patient 1 has 2 spike mutation at the 23,403 zone and 23,603. And patient number 2 is completely different, has 3 spike mutations. So and some patients don't even have a spike mutation. So when you're giving a messenger RNA that's match to that sequence, are you really giving that? And the problem with matching viruses is they're constantly mutating, so you never really catch the virus. Next. We also discovered during the pandemic that if you give hydroxychloroquine, Z-Pak, vitamin C, D, and zinc in 17 patients, 16 out of 17 eradicated the virus, didn't have the virus in their stools within 5 to 10 days. We didn't publish that because good luck publishing anything that has to do with treatment during COVID. But we did notice that one patient still had COVID in his stools persisting, and that was a patient that had full-blown, that had HIV. Next. So if COVID is in the stools, we ask the question, what is it doing to the microbiome? Next. And what we started looking at is family portraits, right? Next. Kid with 0 bifidobacteria, mother with bifidobacteria. The other 2 kids had bifidobacteria. The 3 people with bifidobacteria did not catch COVID. The kid with loss of bifidobacteria had COVID. Next. And they lived all in the same family. We then looked at another family of 6, noticed that out of the 6 patients, the baby had a lot of bifidobacteria, never got COVID, but the other 5 family members had COVID. Next. So we extrapolated this data to become 72 patients. The ones in orange were the high risk exposed, never got COVID. The ones on the left, the 43 patients, had severe COVID. And you could see bifidobacteria was definitely absent in those severe patients compared to the people that are exposed and high exposed and never got COVID. Next. So we published this paper, which was basically the missing microbes of COVID, where we showed loss of bifidobacteria, loss of Faecalibacterium, high Bacteroides, loss of diversity. And that became kind of our beginning. Is that how we can start looking at immunosuppression, immunity? Is that how we can start looking at how to protect ourselves from COVID or the next viruses? Next. So the hypothesis was really, bifidobacteria starts disease. Bifidobacteria, you need to have a certain amount to protect yourself from viruses. What is bifidobacteria? $1 billion industry. Next. So we start, and you could see billions. Next. Who has bifidobacteria? Well, typically newborns have a lot of bifidobacteria. Mom, typically not so much. Next. The process of aging. Next. This is a mom with her newborn, and you could see a lot of bifidobacteria in the newborn. The mom actually got COVID during the pandemic, was breastfeeding her baby. The baby had a sneeze the whole time, never had any symptoms, never had any COVID, basically, and 98% bifidobacteria by the time he was 6 months old. Next. So mothers and infants, a lot of bifidobacteria in infants, not so much in the moms. They just delivered a baby. They're stressed. They have a lot of things going on. Next. The process of aging is really this loss of bifidobacteria. When you start looking at newborns versus old people have 0 bifidobacteria for the most part. Newborns have a lot of bifidobacteria. Next. The other interesting thing about bifidobacteria is it decomposes plastic, and we're going to start seeing a lot of data. So do we have a climate problem, or do we have a loss of bifidobacteria? Next. We noticed loss of bifidobacteria in invasive cancer. Is that why, we're seeing a lot more cancers if we've killed our bifidobacteria, possibly with COVID or the vaccine? Next. We also noticed loss of bifidobacteria in anxious patients. Next. Is that why people with COVID had a lot of anxiety? We noticed it with Alzheimer's. Next. We noticed it with long COVID. So our population of long COVID and vaccine injured, by the way, have the same signature microbiome. So what's going on there? Is it the commonality between a vaccinated patient and a COVID patient is really the messenger RNA? Is the messenger RNA acting as a bifidophage? Next. We noticed it in autism when we looked at kids and siblings that were neurotypical. Kids with autism had low bifidobacteria compared to the neurotypical siblings, and then compared to the rest of the kids was a much higher difference in bifidobacteria. Next. We also noticed it recently and presented it in bipolar disorder. Next. Sorry, there's a lot of data. Lyme disease, next. We noticed it. And Crohn's disease, next. So what increases bifidobacteria quickly? Next. So we noticed vitamin C increases bifidobacteria. We gave vitamin C before and after, noticed an increase within 24 hours. Next. Next, we published on that. Bovine IgG, take a picture of the slide. We call it Biome Booster Plus. Increases bifidobacteria, decreases Bacteroides, increases Firmicutes, decreases Proteobacteria. So if your signature microbiome is that, you may want to try some bovine immunoglobulins to help you. Next. So it's all about signature microbiomes and how to improve them. Next. Ivermectin, the most controversial drug, increases bifidobacteria, but only within 24 hours. Very important finding. We need to do long-standing studies on long-term. Next. Effects of ivermectin. And this is why during the pandemic, you saw me posting on Twitter and X, improved oxygen saturation. Next. So the hypothesis there was next, and published actually the paper that was actually retracted, that we're trying to republish again. But the hypothesis there was really that bifidobacteria binds to TNF-alpha, prevents the cytokine storm, and therefore removes all the cytokines from the lungs and flushes them out of the colon and allows you to breathe because the body is a full motion from the lungs to the colon. Next. Next. The role of probiotics is the label real. Is the bacteria dead or alive? Does the probiotic stop your own ability to make your own bifidobacteria, and does it in plant? This is a very important thing to know. Next. When we looked at probiotics, next, we noticed that probiotics that are not regulated, actually patients have zero bifidobacteria compared to regulated probiotics versus the healthy controls. So maybe there's a problem with probiotics if they're not real or if they're dead. Next. Labels mislead. We looked at 26 samples of yogurts in my area. I don't say which yogurt because I don't want to kill the whole industry of yogurts and drinks. But out of those drinks, 26 products, one of them was $27, by the way, had only 3 of them had bifidobacteria. The others had zero bifidobacteria. Next. So I'm going to touch next. So what kills the bifidobacteria? Next. We discovered messenger RNA. This paper got recognized at the American College of Gastro. Next. So we discovered messenger RNA decreased bifidobacteria by 50% within a month. But then when we looked further in other patients, next, we noticed that it continued, keep going, next, to decrease the bifidobacteria 90 days, up to 9 months after vaccination. So that was a problem that we were alerted to. And now we're continuing to see it. Newsflash, we're seeing all the vaccine injured that are presenting in our offices. They all have zero bifidobacteria, but more importantly, they have zero actinobacteria. The whole entire phylum is wiped. Next. Antibiotics, we know kill the bifidobacteria. Next. And then, of course, next, alkaline water, not a fan. Lack of exercise, lack of sleep, stress, alcohol. Yes, 1 glass of alcohol can improve your BIF. Possibly, but it may kill your BIF down the road. It is a sterilizer after all. Next. And now I'm going to leave you with the last couple slides, healing, understanding the microbiome. This is how I've healed my patients with long COVID. This is how I'm trying to show the data. And we're working with, personnel that used to work in the government to basically validate this, to make sure that what I'm seeing is real and there's actually signals there and not just Sabine Hazen seeing a bunch of N of 1s. Next. So in my world, it's find the bug, creating the problem, kill the bug, restore the microbiome. Next. So we saw with fecal microbiota transplant, that was the N of 1 that showed improvement of Alzheimer's. What is happening with Alzheimer's when we take feces from a wife and improve the microbiome of the patient? The microbiome tells us. Next. Autism, more than hope, a nonverbal 19-year-old says mama after fecal transplant. Again, microbiome manipulations. Next. And you could see this kid had a lot of Bacteroides, Lactobacillus animalis. Next. And the donor was a lot of diverse microbiome, and then he started matching the donor. Lactobacillus disappeared after transplant. The donor didn't have that bacteria. Next. Proteobacteria, had a lot of proteobacteria. That disappeared after transplant. Next. And that's where the microbiome helps us in figuring out what we're doing with improving these kids. And then actinobacteria started climbing. The second column is the donor. This kid improved his actino and thus speech resumed. Next. His CARS and ATEC scores improved. Next. This was a case we did with the FDA, by the way. This is C. diff. What is happening with C. diff is you actually kill the microbiome when you give vancomycin, flagyl, fidaxomicin. The last column is implantation, and you could see you're restoring the microbiome, which, by the way, improves C. diff, and that's why fecal transplant has such a high success of improving C. diff. Next. Diversity is health. Loss of diversity is disease. This is what we learned from the microbiome. Next. But not all diversity is good. This is a patient that started off with a diversity of 3.8. Suicidal, C. diff, chronic UTI, chronic psoriasis, first column. Second column is his donor, 6.2. You could see how he matches the donor and continues to improve. Now we have a clinical validation that basically says this patient is improved. He doesn't have psoriasis, no chronic UTI, no suicidal ideation, no C. diff. But more importantly, his microbiome is diverse and he's looking like the donor. Next. So engraftment is important when you do fecal transplant. The microbiome helps us in determining engraftment. And that's my role on the front line of fecal transplant, on the front line of COVID, is really demonstrating how do we get a cure and what did we change in the microbiome. Next. So we improved the case of Tourette's during the pandemic with SARS-CoV-2 that we found. We eradicated the COVID, improved the bifidobacteria in this patient. Next. And she improved. You could see actinobacteria improving. Next. Tourette's improved. She's a graduate student. Next. 4-year-old appearing with 2 months ago with 100 seizures day and night on rectal depakoid and diazepam due to seizures. Basically, yes, I have 5 minutes. I'm almost done. Next, we noticed that she had low actinobacteria and then started improving. As her seizures disappeared, the actinobacteria improved. Next. Her bifidobacteria was 0 on DNA and RNA. RNA is the reproduction of these microbes to see if they're alive. When she reached improvement and her seizures stopped, and by the way, this kid had 100 seizures day and night. Her dad was a prominent neurosurgeon. He couldn't believe what I was doing on her. But essentially, we showed him the data with the bifidobacteria improving and also her seizures improving. This kid has no longer any seizures. It's been since last July. Next. Her bacteroides dropped. Next. Her Bastiata were adequate for her age. Next. And diversity was adequate for her age. Next. Diversity. Next. Next. And I'm going to show you a case of metastatic. It's a guy with metastatic squamous cell cancer, stage 4, unable to swallow, hemoglobin of 8, weight of 120. Next. His tumor decreased after treatment, refloralization, I like to call that. Hemoglobin went up to 10.6. Tumor dropped to 12 centimeters. He started speaking. Weight went up to 136. Next. Let's look at the bifidobacteria. Actinobacteria went up after treatment. Next. Bifidobacteria went up 14% with tumor shrinking. This guy refused chemo, refused radiation, refused surgery. Next. And his verruco microbiota dropped. He had 30%. Next. Which is acromencia, making him at a regular level. Next. I'm going to show the last 2 slides, 2 identical twins that I'm very excited about. Nonverbal, you could see they had 3 different groups of microbes identical in both twins. At 1 month, those microbes disappeared. 9 months, they completely disappeared. Next slide. These twins in July of 2024 were nonverbal, now are fully verbal. Look at twin 1, a lot of bifidobacteria in blue, and look at twin 2, a lot of bifidobacteria in the blue. Those kids are fully verbal now, fully functional, going back to school. You can't even say they were ever autistic. This is the future that I'm bringing you onto. Next. Save the BIF. I think I made my case. Bifidobacteria is important. Next. If the bee disappeared off the face of the earth, men would only have 4 years to live. Next. What is the bee? The bee in its microbiome has lactobacillus and bifidobacteria, 2 very important microbes that helps us degrade sugar and calcium. Next. What Albert Einstein really meant to say is if the BIF disappeared off the face of the earth, men would only have 4 years to live. And considering that we now have climate issues and everything, and microplastics are decomposed with bifidobacteria, I think he's right. We're on the way to extermination and extinction as humanity if we don't stop the craziness that we're doing of killing these good microbes. Yes, you can kill the bad microbes, but you got to remember to regrow and refloralize and implant. And on this, I'm done. Thank you so much, everyone, for listening.
Thank you very, very much, Sabine. Great. Let's bring in our panelists who will be happy, I'm sure, to quiz you on these questions.
Yes.
So go ahead.
Sabine, I'm still dizzy. if you ever need a job in Cyprus, we could treat 3 times more patients with you as an MD here.
I would love to set up a clinic. I'll be there. I'll teach the doctors, and we'll go on there.
Amazing.
Yeah, they call me Hurricane Hazen for a reason.
All right. Yeah. So first question, fecal transplant versus cotton ball transplant. Advantages, disadvantages.
What do you mean by cotton ball transplant?
In Europe, we have a technology where they don't use the feces, where they transplant, where they kind of clean the feces and they extract and put them in cotton balls.
Yeah. And they basically are giving the metabolites, right? I don't know. We don't have that much data. And here's the problem why we don't have that much data is because, listen, in America, we're limited. You cannot do fecal transplant unless you have C. diff. And unfortunately, you cannot understand these microbes unless you have a validated assay to look at these microbes and you have a proper protocol that you can kind of like is similar that you can say, okay, well, this is why I'm accomplishing something, right? My goal and my hope is to work with all these companies that are doing fecal transplant and to say, let's look at the microbiome. You know, Dr. Barodi and he was doing fecal transplant in Australia, and I was looking with him. And so we started understanding what is a normal microbiome. But here's the thing, you can't really understand what is a normal microbiome until you've analyzed a lot of stools with a validated assay, and you kind of say, why is Alzheimer's looking like this? There's a signal in Alzheimer's. There's a signal in Parkinson's. There's a signal in a carnivore. There's a signal in a vegan, right? Because like I showed at the beginning, you cannot compare 2 samples. So if I'm seeing a signal when I'm improving, what I'm showing you, which is really, you know, beautiful and all that in my own way of presentation, is also very skewed because I'm seeing what I want to believe of bifidobacteria, right? But there's so much more to the story than bifidobacteria. Bifidobacteria was my discovery during the pandemic where, hey, if I maintain a certain level of bifidobacteria, I'm okay. I can be with a bunch of patients with COVID and I'm not going to get, you know, because I tested my stools the whole pandemic. My case has not come out yet. You know, we're having a lot of problems with retraction of papers and journals. And I think all that is coming to light where doctors are saying, wait, why is this paper retracted? Finding COVID in the stools was a landmark paper during the pandemic. You know, it's what got the National Institute of Standards to also look at it, but we were looking at it clinically. Why was that retracted? Isn't that important to have a marker of COVID in the stools? And why couldn't we ever develop an assay? Here I am doing fecal transplant. The reason that I developed this assay and looked in the stools, I didn't want to give stools with COVID or for that matter, spike protein to a patient. So for me, it was so important to make sure my stools had 0 COVID, had 0 spike protein. You know, I worked with Kevin McKernan. I worked with Philip Buckhorst. I worked with David Speaker. You know, I brought David. I flew him in from Toronto to look at stools for COVID, to look at, you know, vaccines, to see what they were seeing, because I wanted to see it with my own eyes. It's important to validate. It's important to be righteous. You know, I'm now working with someone from that used to work at the government. And I said to him, I don't want you saying that Sabine Hazen is doing a great job. I want you to show me where my flaws are. What am I messing?
Sorry to cross you again. Coming back. So you think the reason why some of the patients that are very, very ill, like severe long COVID, they didn't respond that well to fecal transplant because it was spike or COVID in there?
Possibly, yeah, you're not testing the stools. Yeah, you're not testing the stools.
Well, they
And
didn't respond negative to the cotton
Well,
ball procedure.
yeah, so let me tell you, there's a couple problems with fecal transplant, okay? One, it's great that you're doing fecal transplant. By the way, those cases were not fecal transplant. I want to assure everyone. This was a manipulation of the microbiome, understanding what kills the microbiome and what increases the microbiome, okay? There's a lot of drugs out there that are increasing the bifidobacteria, okay, that nobody really knows. We know because we've done the research, okay? So this was not fecal transplant. This was really a consortium of microbes, okay? This was really innovation at its best. Kind of what I showed with COVID, where you killed the virus with hydroxy and Z-Pak, you increase with vitamin C, vitamin D, and ivermectin, the bifidobacteria, and now you have the patient restored in a way and no longer having COVID or long haulers, right? Here's the problem. When you don't have an assay to kind of monitor that, you're in the dark. So fecal transplant, the reason it didn't work is because are you putting your patient back into an environment where first of all, did your patient have COVID or was vaccine injured because he had a dysbiosis to begin with, right? So many of these patients, when you take a history, they say to you, well, doctor, I was gassy. I've been gassy my whole life. I've had diarrhea before COVID. So when you take that history, first of all, if they're depressed, they have a gut dysbiosis. If they're anxious, they have a gut dysbiosis. Bipolar, gut dysbiosis. So if they tell you anything from anxiety to bipolar disorder to Parkinson's, they had a gut dysbiosis. So you're not treating long hauler. You're treating the gut dysbiosis, okay? Now, here's the problem with treating the gut dysbiosis. They started off with a gut dysbiosis. Until you know what the gut dysbiosis is of Parkinson's or Alzheimer's or autism, you're not going to know how to fix. So you could put stools, a new microbiome, and hope that you're going to improve. It's like putting mud on the wall and expect it to stick. But until you see that engraftment, you are not going to reach improvement. The other thing is, are you putting your patient back in an environment of mold? Is your patient exposed to a wife that carries a bunch of microbes that she constantly is feeding him? Remember, when we treat Giardia, we don't treat Giardia one patient, we treat the family, because chances are they bounce off of each other, right? So yes, you can change your whole microbiome, but if you go back to the environment, if you go back to the people you're around, if you go back to a lab where you're exposed to multiple microbes, you're going to get yourself back to that dysbiosis. This is why these fecal transplant are not helping. Second reason they're not helping, yes, you're putting in microbes that probably are incompatible with your patient, right? Should we be putting microbes of an Amazon woman in a New York City girl? I don't think so. I mean, that microbiome is full of diversity. The microbiome of, I'm looking at South Africa, and I'm sure, I'm going to have questions on South Africa. The microbiome of South Africa, and I was in Zimbabwe, I took microbiomes of people that are in the jungle with the elephants and the zebras and everything. The microbiome is completely different than my girl that's in California, that's in a sterile environment. Am I going to give that microbiome to my sterile girl? I personally think that's ridiculous. That's dangerous. You've not accommodated to those microbes, right? I mean, I went to North Africa and I got E. coli, and I'm from North Africa. I'm from Morocco. But here's the problem. When I moved from Morocco to America, I diluted my microbiome. We have globalized humanity. Our methods, our foods, our drinks, is being copied. I was mortified when I heard the Amazon jungle is getting Starlink, and now the kids are getting foods from America. Are we changing the microbiome of the Amazon jungle because they're now adapted to this civilization of looking on TikTok at people eating Doritos? This is scary to me. So my whole mission is not really just, hi, I'm fixing long COVID, autism, Parkinson's. My whole mission is to preserve the microbiome of humanity, preserve Africa, preserve China, preserve Japan, bring back America to some kind of, health. We were the worst during COVID. We need to learn.
Sabine, speaking about preserving, what do you think does the spike, the wild virus, and the mRNA cause in the biome?
I think it causes the bifidobacteria. I think it causes the bifidobacteria. I think you put the spike in the blood. Again, this is my hypothesis, my vision. There's no, I don't want scientists going crazy on this. It goes in the blood, travels the blood, and then basically goes in the stools and pops the microbe. Okay. So, and it is there is a principle of bifidobacterial. We use viruses to open bacteria, to kill bacteria. So that's what I believe happens.
And before
Are
we
you
move
familiar
on,
with
just
Calabronia's work, actually?
I'm sorry?
Are you familiar with Calabronia's work where he
I'm
showed that
actually
the
working.
virus...
Yes, and I'm actually working with him, and we're going to collaborate. We're on a WhatsApp. Believe me, I picked up my biggest thing during the pandemic is I picked up all the warriors that were righteous, and I brought them on to Progenobiome, not because I need to develop a consumer product or consumer lab, mainly to have that brainiac. You guys stood up during the pandemic and you're courageous and you want to see the truth. You're hungry for the truth, like I am. And you're going to be a disruptor and you're going to be shaking the beehive of pharma and the narrative because you want to see the truth. We cannot be biased in science as much as we are all biased in a way because we want to see our science grow. We cannot, because ultimately it's going to affect us. You cannot cheat science. You have to call a spade a spade. And then you have to give your data and say, can you reproduce what I saw? Are you seeing what I'm seeing or am I full of it? Right? That's why I wrote the book, Let's talk shit, I mean, am I full of it or do I see something? And I don't mind stirring it to see that something because that's my longevity. That's my truth.
Okay. Before we move on, Shankar, do you have a question before we bring in
No, Philip. I just wanted to reiterate the importance of Sabine's work. I think we live in a biologic soup, and the microbiome that surrounds us, that's part of our ecosystem, we are an ecosystem as well. And I think Sabine's correct. We've had Chinese people eating a Chinese diet all their life, and the microbiome helped them adapt to that environment. Now the guy's eating a Big Mac burger in the States. His microbiome is going to change. Yes, our microbiome helps us adapt, and it's imperative for our survival. But I think as well, the decimation of the diversity of our microbiome is the problem. It stifles our ability as helical DNA creatures to adapt to our environment. And I must compliment you on trying to preserve it.
Thank you.
Excellent. I tell you what we're going to do now. We'll hear once more from our sponsor, and then I'm going to bring in our other speakers. And then this is where we have our roundtable discussion, where I have my questions. Anybody else has questions to anyone else, it will allow us a chance to discuss. I'm keeping an eye on the messages and the comments, which are coming in thick and fast. So yes, please keep going. But let's just quickly hear a message from our sponsor again. Wonderful. So great. Thank you, everyone, for being here. You may notice on the screen, I will sometimes bring up the comments so you can see them real time. Many people are asking questions. So I guess my first question, after listening to the range of the discussion, because we had a discussion covering the mitochondria, the way how the spike protein damages the mitochondria, the way how the spike protein interacts with the clotting process, and now the way how the spike protein interacts with the microbiome. Have we ever seen anything like this before?
Definitely not, Philip. We've never had a virus that infects human beings and bacteria. Simple as that.
I also think we didn't have the technology, I mean, this technology of looking at the microbiome is fairly new. So maybe we did, we just didn't know. But definitely this was a manipulated microbe. I think we can all agree on that. We're in the world of designer bugs now. They can take a bacteria and a virus and put them together. And now you have a super bug. And so we're constantly fighting these super bugs.
One of the questions I wanted to ask Rezia is, when you look at the changes with the microclotting, which seems to be persistent, meaning that it's not as if it disappears. One of my thoughts was that it is quite likely that the driver is the microbiome. Any thoughts on that?
Yes, already in between 2011 and 2016, long before this virus that we see now, we have shown that LPS, so the outside wall of bacteria, and LTA, so from gram-positive and gram-negative bacteria, can actually cause microclotting to happen. So our thoughts at that stage was that if you have got gut dysbiosis or even dysbiosis of your mouth, in the case of P. gingivalis, which is a mouth bacteria, and it gets into your body somehow, that if it travels into your body due to dysbiosis and it's shed in your body and in your circulation, then it can trigger microclotting. So we do know that in our hypothesis many years ago, before the microbiome was trending, the term microbiome was trending. At that stage, people were saying that there isn't a blood microbiome. We were saying that all these parts of the pathogens can interact with a direct protein-protein interaction and can cause microclotting. In fact, there were a few publications many years ago already that show that there are LPS and gingipains, which is from bacteria, inside the brains of people with Alzheimer's. So it's, I don't think it's a new concept. As Sabine mentioned, is probably not. There was probably not many techniques to show it or to test for it, but definitely there's significant interaction, and still is, without even virus without you having contracted COVID. If you've got some viral or bacterial post-viral disease, even like ME/CFS, I think you've got a possibility where bacteria and viral products in circulation can attack your coagulation system.
Well, it kind of leads into another question for Bridget, where if we can see that spike protein can interact with human cell physiology and damage mitochondrial function, is it feasible that we're seeing a similar thing with other microorganisms, bacteria, where their mitochondria are damaged, and this may be a mechanism where we see a connection across the board?
Yes, it's not only SARS-CoV-2, not only the spike protein. We know examples that, for example, herpes simplex virus, Legionella pneumophila, and other bacteria interact with the cell and with the mitochondria at different stages. Also, the one with the ATP production, the other with oxygen radicals production. In the difference, the SARS-CoV-2 spike is really doing it different, simultaneously at different stages in the cell. And but if I refer to the hyperactivity of platelets also have mitochondria, and we did a lot of with platelet mitochondria also in with platelet-rich plasma and so on, with a little bit with facelifting, and we looked at the activity of the mitochondria, and it is really dependent how they react to platelets. It depends on their mitochondrial function. Yeah, so it could also be a link. Perhaps also another link with the LPS and peptidoglycan and other bacterial microbial metabolites. Yes, LPS, we did a lot of studies years ago, also 20 years ago. And we, you know that LPS, for example, is going with toll-like receptor 2 and toll-like receptor 4, and they have different toll-like receptors. But the cells or the immune cells are also able to produce lipopolysaccharides binding protein, and then which goes to CD14, and then the macrophages can remove the LPS. But if the immune system is not functioning, for example, by mitochondrial dysfunction, but other things, then the LPS and other metabolites can't be removed, for example, by LPS binding protein, which is produced by cells. And so it is an interactive or a problem which is not one problem, but it's interrelating or interdisciplinary problem.
Yes, and I was thinking, Marcus, is that when you guys are working with apheresis and you are removing, in a sense, microclots, you're removing autoantibodies. What seems to be interesting is when you remove it, the patient may improve, but they reform these patterns over time, so they have to continue having it done. What is it that you guys see in real time when you have taken out a lot of these inflammatory patterns?
Excellent question. So yes, as a mono treatment, as a standalone treatment, apheresis alone, no matter if that is HELP apheresis, inosteresis, or immune absorption, or a combination of the three, can only go so far. If you don't combine it with either hyperthermia, nutraceuticals, especially molecuzan, or other therapies, where you also go after the root cause, apheresis would probably work for 40, 50, 60% of the patients, and they wouldn't relapse because they're going back to homeostasis and the body then can take over and recover. But for the other patient group that you just mentioned, multimodal combination therapy has been the only way that we saw working to achieve permanent results. And then there's a third group of patients. So let's say you come in a wheelchair, we bring you up to 65%. That is a great result, right? But then they come back and go like, what about the other 35%? And they come a second time, and we then add monoclonals in, monoclonal antibodies, IVIG, subcutaneous IgG, peptides. So literally, if you don't tackle this problem from all sides, your cards are quite bad. So again, there's a lot of monotherapies, but in our experience, if you don't combine therapy, sometimes even at the same time, you just don't have the results that you get by combination therapy.
Well, this leads right into Shankara from a clinical point of view. How do you know who needs what? Because you can't do everything to everybody. From a clinical point of view, what do you think?
Philip, depends what you're looking at. If you're looking at COVID, we know that it triggers microclotting and immune dysregulation. That's easy to pick up if a patient progresses into illness after seeing signs of early recovery. They remain unwell for a long time, you know where to look. When it comes to the vaccine, again, the same thing, immune dysregulation and coagulopathy. And I think clinically, it's easy to tell from without testing what a patient's presenting with. Is there eczema? Is there atopic symptoms, asthma, gut problems with allergy? Sometimes allergy can affect your nervous system as well. So I see a lot of benefit in immunomodulation with antihistamines and a steroid to suppress and restart the immune response. With coagulation, you see patients' deterioration in eyesight, symptoms of peripheral neuropathies, those kind of things. So symptomatically, you can basically, if you're looking for two things, you know where to look. It's those that don't respond to treatment that need further investigation. Just feeding into what Sabine was saying about the microbiome and how it influences clotting and with Resia, and the lipopolysaccharides Bridget was talking about and how they can trigger these microclotting events. Is it that with other pathogens, they were very specific in how they dysregulated mitochondrial function, affecting a single pathway, and we always had other means to stem it. But because spike protein has such a diversity of mechanisms blocking apoptosis, interfering with interferon responses. It actually covers all the mechanisms that the body has to rectify itself. And that's why it's become a very persistent problem, which feeds into the fact that this is not a type of bug that we've seen before.
Yeah, I think that this is part of the problem, is that we are seeing pathology like we have never quite seen before. And I think that one of the things I found most concerning was, as Sabine had said, that research that didn't fit in the narrative was suppressed. And I think it was because the scientific community thought that they had all the answers or all the mechanisms. And now we are in a situation where we are having to start from scratch 5 years down the line when disease has evolved. And it is very difficult to play catch up to a condition that is continuing to evolve. And so we have a really serious situation where it's not easy to do this kind of research. We are appreciative to say, like, for instance, Rezia, who was doing this microclotting research beforehand. That's unusual in most aspects of the pandemic. For many other things, we don't have a lot of upswing in terms of the science. Does anyone want to explain how we managed to get ourselves in this trap?
Censorship, Philip.
The problem,
Suppressing the doctor's lounge.
I think we played with something we shouldn't be playing with, and that's taking a virus and a bacteria and putting them together. I think, we opened Pandora's box when we started doing that. I think we didn't know, the messenger RNA may have some benefits, they thought it was safe, but maybe it's not. Maybe the spike protein itself, I have a belief that it probably activates viruses. One of the things I do with my long haulers and with my long COVID and my vaccine injured is actually I test herpes, EBV, HPV, I mean, not HPV, EBV, JCV, CMV in the blood. And inevitably, these people all have positivity in their blood for these viruses, and they didn't have them before the pandemic. So did the spike protein activate these viruses? Is that why we see CMV encephalitis? Is that why we saw herpes encephalitis? Is that why we saw shingles? Is that why we saw hearing loss after the shots? We've got to ask these questions, because if the spike protein is a loose cannon that has an ability to penetrate a virus and activate it, or bacteria, how many patients developed C. diff after the shots? How many people developed H. pylori after the shots? Is it activating a bacteria? Is it activating a microbe? I think this is something nobody anticipated. And, to go back to the mitochondria and to go back to the bifidobacteria, in my line of work, bifidobacteria helps decompose sugar. Without sugar, you don't have your Krebs cycle doesn't work. Your mitochondria is not working. So, this is important to understand.
Well, here is a point that somebody said that caught my attention. Tired of this focus on pathogenesis. They want treatments. They don't care that you're searching, or they want to get better. What's the answer?
Well, I think invest in research first. That's actually done by people that are righteous. You have the team right here. Invest in Professor Razia's research. I'd love to work with Bridget to kind of see, if there's a spike protein in the stools, for example, etc. I think that's the first thing. We need better. I know it's frustrating to people, but we need better diagnostics. Okay, because we've not cured anything. Parkinson's is highest than it's ever been. Alzheimer's is the highest it's ever been. We need to force our governments all around to invest in proper research that's non-biased, and to have a team of collaborations across the world that's non-pharmaceutical driven, and not just to develop a product. And also utilize the drugs that have combination. For example, working with Dr. Shanka, basically, to say, what are you using? Let me see what it's doing to the microbiome. Dr. Cypress, same thing. What are we doing? So I think to shoot out a treatment, I will tell you that I have a treatment that works for one person, but it doesn't work for another person. I need myself in my lab to figure out. So I have the ability to go back to my patient and ask the questions like, why didn't it work? And then I formulate a new formula for my patient. It's precision medicine, but it's precision medicine that needs new tools to look at the microclots, new tools to look at the mitochondria. What we have is not helping us.
Well, let me come back to you, Philip, on our point of view. So what we've done, I currently have the feeling I see Brigitte more often than my wife. So we're working very closely on so many things. There's so much data interchange. Everything from science goes directly, at least here in our clinic, into treatment. So science-backed only, believe in the science. Second of all, don't hesitate, invest, create treatments. So literally, we started with a portfolio of one treatment. That was HELPAparesis 4 years ago. By now, we have a portfolio of over 30 treatments, right? And for me, it was very important to collect all of these treatments that are out there and test them. And the ones that didn't work for our patient population, we no longer offer them. Of course, everything has to be done under the do no harm principle. But there is a lot of treatments. Now, is that enough? Well, how do we know which treatment we use for which person? Now, in my opinion, we're already at 40 to 60% precision medicine. The tests of Brigitte, the tests of Risha, other tests, GPCR autoantibodies, the whole panels, the new labs that are now coming up. So there is a big part of the treatments that we can already touch it very well. But let's be very honest. For now, we're also having a part of the treatments where the good old pattern recognition. This treatment worked for a person with this symptom set. Let's try the same for the next person with the same symptom set, where that also has to be applied. And we're not shy of saying exactly, this is what we know, this is what we don't know. So here we are in the section of precision medicine with the treatments. Here, it's good old-fashioned pattern recognition. We don't know what's going to work for you. Let's throw things up at the wall and see what sticks. That is still a part of our clinical reality.
And when you think of it, Bridget, from your point of view, looking at the persistence of the spike protein, one of the questions would be, is that, does that necessarily mean symptomatic disease? Just because someone has significant levels of circulating spike protein, whether it's in immune cells or in exosomes, does it always equate to significant disease?
No, that's right. But the spike protein is a pathogenicity factor, and a pathogenicity factor should not be in our body, independent from which microorganism, from which virus it is. So it depends on the individual person. If she has chronic diseases, or whether she has, or he or she has a good mitochondrial performance, then they can combat the spike protein, but not forever, but for a short time. It depends, when did we test it? Did we test it 4 weeks or a few weeks later again? Or we don't know exactly whether some spike are still in the body, but it's not coming out because the immune system is not fighting. But your question is, or the question was, could somebody has spike proteins and he's feeling well? Yes, I have also spike proteins. I had not much, but I was feeling well. But it is you have to try to destroy them and to help your body to combat this this pathogenicity factor. So it's important to know, and then to decide. Yes, you have to support your body.
Yes, I think that one of the things that I found looking at the research, Rezia, is that looking at the characteristics of the clotting, and it's not just the microclots, but also the macroclots, some of the associations with the embalmer's clots and very unusual patterns. And it does seem to be primarily immune related in terms of the speed of onset and so on. So people may have microclotting occurring, as you said, in almost 100% of the long COVID patients, but it doesn't mean that they will necessarily progress to having macroclots. And there's clearly some kind of transition. And this is where I've been interested in what I call the COVID storm. Storm meaning spike triggered autoimmune response mechanism. So in the person who is immune primed, and it could be primarily in the vaccinated, but it could also be in the unvaccinated, then has a reinfection that triggers the immune system and then causes what I think are the nets, not actually nets, but METs, macrophage extracellular traps, to then coagulate to make big clots. Any thoughts about that?
Yes, definitely. We have been noting nets, so the neutrophil extracellular traps, in many of our microclots structurally associated with it. Definitely, I think it's not a one scenario fits all. It's not only having microclots. It's not only having platelet hyperactivation. It's a multiple or a huge combination of systemic factors playing into this disease, and then a systemic endothelial activation of endothelial damage that causes these symptomatic symptoms that we see in many of the patients. I do agree with you, Philip, that multiple reinfections or then vaccination and then an infection, we simply don't understand fully what triggers what place when. We simply don't know yet. And as Marcus said, that it's not a one treatment for everyone. Every single person is different. And I think, just circling back to one of your previous comments, I think the important thing why we don't see adoption of new technologies or new concepts is simply that people follow a process where they learn something at medical school and they now want to just use it as is for the next 50 years that they practice medicine. They're not open for change or different opinions or other methodologies. And it's really significantly difficult to break into the medical and research fraternity with a new concept. There's something that I learned from Doug Kell, my long-standing collaborator. He always says, in this environment, and it's very true for COVID research and COVID treatments, is the old concept of when people don't understand it, it doesn't exist. They will say it doesn't exist. Microclots don't exist. They're not there. Then the next phase will be, they say, oh, it exists, but it doesn't mean anything. And then in a few years' time, we will probably be sitting here and we will be saying, Sabine's work, Marcus's work, our work, we knew it all along, so it's nothing new. So it's the phases of adoption of not only the medical fraternity, but we are now in that in-between phase. I think we are now, people are believing that there are something like microclotting. They want to term it something different, which is possibly that. The same with Sabine's work, the same with Marcus. Should we do help apheresis? I think we're away from it where it doesn't exist. It's not a technique to use, to now it works, but perhaps not 100%. So I think it's our responsibility as researchers and with forums like this to just chip away one by one, put out the research, argue against what people say, argue with them, discuss with them terminology, methods, treatment, listen to all the sides. But then we would want to have an opinion that we can all say something and we are listened to, and some things might turn out to be wrong. Sometimes things are right, but an open mind. And unfortunately, in medicine and in research, some don't have open minds.
I'm going to have to throw in this very difficult question. You don't have to answer this question, but I just noticed it in the comments. Look at it, reflect on it, and see what you think. This is from Tom. If the spike protein from the virus or the vaccine is the cause of all this damage, does it make sense to immediately stop injections for everyone?
Yes. Philip, if you look at what we're doing, the spike protein it biodistributes with the vaccine more than with the virus itself. We're sitting with a very complex problem as we see it, right? You got the virus, you got the vaccine. The virus can infect microbiome. So when you look at just the microbiome, you can get the virus infecting the microbiome and resulting in a cytokine storm from that infection of the microbiome. We've seen the diversity from Sabine of microbiomes, but we've seen the similarity in families of microbiomes. So is that the genetic predisposition? Is it the genetic predisposition of the genetics of the microbiome that we all have that's actually making us respond differently? So you got the spike protein influencing the microbiome. You got the damaged, persistent spike damaging mitochondria, creating a cytokine storm as well. You've got, with Rezia's work, the microclots that can trigger the same issue with exercise, releasing cytokines. So everything seems to feed into each other, from the microbiome to the persistence of spike in the cells, to the immune dysregulation. So everything is interconnected. And so we're going to have to look at how to sort it out on either phenotype or blood groups, or who's more predisposed, which bacteria. Because, I mean, the lung conditions that we saw in the first wave, could have been, it was cytokine release, but was it cytokine release from infection of your respiratory microbiome that triggered the entire process? Or was it the spike protein that actually triggered the immune response directly? So there's a lot of different things going on. I think the right testing is vitally important, and the right segregation of patients as we see them.
I'm going to say something on this point, and you can criticize me on it. One of the lessons that I learned from observing the early part of the pandemic was the use of force to force people to do something.
Yeah, I think that that was everybody I talked to said, well, I didn't want to do
You know,
it, but
we have to.
I was forced to
Yes, we
do it.
have to. Yes, we have to respect the opinion as well, because the truth is that certainly from a scientific point of view, with regards to the impact of IgG4, just that alone from the mRNA technology is technically immune suppressive. So it is likely to have an impact on the severity of the disease. Now, it could come with complications down the line, but if a percentage of the population will never use horse paste, as they say it, or any of the recommendations, say, Sabine has come across, and they will theoretically go with nothing or this, what do you do? I think it's a very nuanced situation. Mhm.
I stand on the path of divided, right? And I don't answer this question with yes or no. Here's why. Science is about asking questions. We shouldn't be going to the right or to the left, and we should give freedom of choice for people that if they cannot go back to work because they're so fearful of the virus, and a vaccine will get them to go to work, well, be it, take your chance. We're well aware, okay? However, we have to convince the other side. So when they tried to conquer, they conquered by dividing the world. We've been divided as humanity into those that believe in the vaccine and those who don't believe in the vaccine. Our job is to convince those that believe in the vaccine that there's a problem, right? But how do we do that? We can't do it by using their methods of, oh my God, shut it down, and this is crazy, and this is not. We got to bring them slowly to kind of say, look, this is what we're seeing, and educating constantly. So, what do you do with the person that's 300 pounds that, doesn't have any bifidobacteria, that is not going to comply and take vitamins, that doesn't have Dr. Shankar or Dr. Marcus or myself as a doctor, who doesn't have ivermectin or all these drugs? What do you do with that person? Do you let them die? That person never got COVID and believes that the vaccine helped her, right? So there's two populations. There's the population that is the athletic, that are very driven and take their vitamins, and they're disciplined, and there's the population that's eating McDonald's French fries. I think we need to bring everybody to the stage and say, how do we safely delivered this technology? And is there a better way to vaccinate? Because think about it, they've basically killed this technology is a new technology. There were other ways to vaccinate that we did not. The vaccine is a brand, right? Let's remember that. So there were other ways. There were also other ways to say, how do we give the vaccine and maintain the microbiome? Or how do we give the vaccine and make sure we don't attain clotting, right? Nobody looked at that. They don't even want to look at the data from Dr. Rezia or Bridget or myself. That it needs to stop. That needs to be coming to the table. And we need to come to the table. That was the problem with the pandemic. Those that argued against the vaccine were never allowed to speak their mind. It was line up or you lose your job, line up or you lose your license. How many of us got tormented this pandemic because we wanted to stand for freedom of choice? And that's what it's all about to me. I believe freedom of choice, make an informed consent, decide.
But isn't a very interesting question not about the vaccine only, but early treatment versus vaccine? That is a very interesting question, isn't it?
They didn't want to hear that hydroxychloroquine killed the virus, right? But they didn't want to hear, but also I came to find out hydroxychloroquine also killed the microbiome. So it was not a solution for all, right? They don't want to hear those because nobody's taking on the research to do these cheap products. Think about it. I stepped into the pandemic with 3 protocols on hydroxy, Z-Pack, vitamin C, D, and zinc, and ivermectin, doxycycline, and zinc with the FDA watching. That was, over $1 million of my savings on that research. And then the work of it, and then bringing money from Dr. Barodi and people that really believed in us. That was not cheap. And we couldn't even recruit because they censored us. Because this was a protocol that was given in full transparency. Think about half the drugs on the market. You have no idea what you're giving. What is Paxlovid, right? Does it have ivermectin in there? We don't know.
Can I also just come in here? We could talk about the vaccine and everything for a long time, but what we must also remember is there are currently very few people are taking the vaccine, I think, but moreover, I would like to focus on reinfections. We are not testing whether children or adults or if they're sick, we don't even know. They don't even know they've got COVID because we don't test. In South Africa, I can say we don't really vaccinate either anymore. So we did, but we don't. So we must really also move further along than just vaccination. We must really encourage governments and clinicians and schools to look at reinfection processes, because we are sitting with a lot of people having acute COVID, but they don't even know they have it. And whether they're now vaccinated or not, it can play a role, but we're simply totally ignoring the current circulating viruses. And long COVID is happening as we speak. Every one of us is just one infection away from long COVID. You never know how your body will react on a viral trigger. You don't know how the COVID infection will react to other viruses that you might be exposed to. We don't test. If someone is sick in a school, and I know there's a whole lot of clinicians in the UK, Ray Duncan, Benita Kane, fighting for clean air in schools because as children are just exposed constantly to this onslaught of virus. So looking at the vaccination is one thing, but definitely the reinfection rate, people don't even believe there is, circulating COVID anymore. The general public don't even realize that they might have had they might have a COVID infection, and they might be spewing out virus to everyone around them, with someone in their near family or close friends might end up with long COVID. So I think that's also a very important conversation to be having is what are we doing about reinfections? And if we don't look at that, long COVID will be a problem forever and ever, because we might be sitting with people now suddenly developing cardiovascular disease, healthy young people developing Parkinson's-like symptoms or Alzheimer's in their 30s. We are exposing a whole population of individuals, young individuals, that will have cardiovascular disease in their 10 years or so, in their 25, 20 or 30. And the conversation is closed. Nobody talks about that. And that's a major problem for me, even worse than the vaccination dilemma.
Listen, folks, I think this is brilliant. And what I want us to do as we close out, we're coming to that 2.5 hour mark. So I want everybody to think about what would their takeaway point be? What is it that they would want to say to our listeners now? I mean, we have almost 570 people live with us now, and more people will watch it down the line. What point would you want to give them based on your research, based on your experience? The thoughts. So we'll start with Shankara, then Marcus, then Bridget, then Rezia, then Sabine, and we'll catch your thoughts as we close out on this conference.
Philip, I think a takeaway point, we are dealing with a bioweapon and it's been made to injure. So we should keep our eyes on everything. What the group here is trying to do is collect information, not for our own patting ourselves on the back, but to inform patients. And I think what patients need to understand, we have a circulating pathogen that was designed as a bioweapon and it's still very deadly. When it comes to the vaccines itself, we're trying to figure out what exactly happened so we can give patients informed consent so that they're well informed, and we're trying to find the information to inform them. I think what people need to realize during the pandemic, it wasn't there wasn't informed consent, there wasn't uninformed consent, there was misinformed consent. And so it was driven to misinform people to get their consent. So I think the information is vital in providing informed consent to everyone. And the principle of first do no harm, when we talk of the vaccines, if they're doing harm, they need to be pulled. Whether the person is deluded in thinking he's going to take the poison, I'm not going to be the one supplying him with the poison because I'm the one that's in charge of his health. So I think it comes down to the product rather than the patient's preference.
Excellent, excellent. And Marcus?
Well, my takeaway is very simple. My message is don't lose hope. I had severe long COVID 5 years ago, so I know how it feels to be looking at the ceiling 23 hours a day, being completely in pain, not being able to talk or even hold a glass of water. And me, and a year later, my mom, we recovered, and so many of us recover. So there is tireless people who work behind the scenes. There's new laboratories built. There is AI integrated. There is new tests developed. There is constantly new treatments. So yes, it is difficult, but there is some things that you can do at home. Inform yourself, reach out to the people that know how to treat it, right? And most important, share your story, because if we don't speak out, nothing's going to change.
Excellent. And Bridget?
Yeah, I have 3 messages. One message for other scientists. We have to be open-minded, as it was mentioned before. We have to talk with each other, and we have to work together. My message to the therapist is, the therapeutics and medical doctors is, we have to go from two sides. I learned it. From one side is from the science, but we need also the experience from the medical doctors. We must know what is helping, and then we have to think what could be the mechanism or the pathophysiology. So we need this interaction. The third message for all others is, yes, there is hope. And I have to agree to Marcus, what he told, inform you and take care of your body.
Excellent. Thank you, Bridget. And Rezia?
Yeah, I think the important thing for me is that we can never lose hope, that there will be some sort of respite soon. I think there are researchers and clinicians working around the clock that don't sleep, that are constantly thinking of how to plan new research, how to look into new frontiers. So for the patients, never to lose hope, because there are people looking out for you. And then I think the most important thing for a message to me is protect yourself. If you're on a flight, wear a mask. If you go out to a function where there are hundreds of people that you don't know what their infection status is, protect yourself. That's the only thing that you have in your hands to try not to get reinfected. We are only one infection away from long COVID, every single one of us. And we don't even know what the underlying effects is. Even if you've got an infection and you are asymptomatic, down the line you can develop long COVID. So I think the most important message for me is protect yourself and look after your family.
Wonderful. And Sabine?
Well, I'm gonna say, as the girl that basically went on airplanes without a mask and somehow managed to survive, I'm gonna say calm down your body and stop panicking and see the light and see the hope and see the lessons we've learned from COVID, the lessons on the microbiome. And I think the microbiome to me is about, calm, zen, decrease stress, decrease anger, decrease caffeine, decrease alcohol, decrease the stress, decrease the fear, decrease the anger, and stop the division. Unite to talk together, bring on discussions as scientists to see both sides of the narrative, not just one, always trying to remain as unbiased as possible so that we can advance science, because it doesn't help us if we fake the science or change this or trick the science. It doesn't help us. We're eventually all going to become patients. To me, it's about a mission to preserve the microbiome of humanity. We've killed too much. We need to build now. We need to bring back microbes. And I think with that is really peace, calm, unity, stop the fear, and go play in the garden. It's summer. All right. That's my message. Thank you.
Wonderful. Thank you all very much. And I know we've got a lot of people with us today live, and I know a lot more people will watch it afterwards. Remember the science. Let's all work together to make a better future. I'll ask my guests to stay with me until after the outro, but for everyone else, have a great evening.